Today, we are going to present some fundamental parts of the cell biology. As you know, this is the main backbone subject of the biotechnology. And today we are going to highlight some key points concerned with the cell biology. What is the cell biology? What is the utility of cell biology in the basic science, and particularly in the biological science? Because as the name indicates, cell biology is the study of cell organisms and the cell. And this is a very important thing. If you go and if you highlight the whole nature, in the nature, there are the very active diversity among the individuals and different types of cells. If we talk about the diversity, because in nature there are some fundamentals things is very important, which is made up of the cells. And what is the significance of the cell in cell biology? Because cell is the basic structure and the function unit of the life. And this is the important thing that which is concerned to the biosphere and the cell. What is the interaction between the gene level of the cellular biology and what is the this is the linear equation which is concerned to the gene to the biosphere. Because if you talk about the fundamental things between the genes, that the unit that is made up of the different cells, and all cells and all tissues are made up of cells, and all organs are made up of different tissues, and this constitutes a different tissue level systems which constitute the whole cellular body. And this is the some fundamental slide which is indicating the original evolution of cells and subcellular structures. Because if we talk about the on the behalf of the structural level and the functional levels, this cell biology discipline plays a very significant role to distinguish the different cells which is divided into different levels of organization. Origin and evolution of cell and subcellular structure. And this is the field, this is the map. So firstly told us about fundamental evolution of life, and particularly this is the self-replicating cells and concerned with the fundamental unit of life plays a very significant role for the organization of the different organisms and for the making of different tissues. And this is some highlights which indicates the significance of the cell. For the first thing, fundamental unit of life, self-replicating structure, and the first cells were observed and named Vallabhbhai in 1665 from skies of God. This is the historical period in 1665. Vallabhbhai firstly named the cell. And some organisms consist of the unicellular organisms, other are the multicellular. Because this thing is very important, because the cellular level is distinguished between the different diverse forms of the cells. If you talk about the bacterial cells, which is unicellular, and if you talk about the multicellular cells, that is the part of the whole protein, eukaryotic cells. Because if you talk about the unicellular organisms, bacterial cells are the unicellular organisms, and this is the average diameter. This is the average diameter 2 to 120 micrometer. This is not a fixed measurements, but it varies, depends upon the occurrence, structure and function of the cell, occurrence, place of the cells that exist in the nature. This is the general average diameter is 2 to 120 micrometer. And this slide is basically highlighting the basic unit of life, structure and function. We already talked about this was the name of again the name of the renowned scientist. We cited this one. He firstly distinguished between the biodiversity among the animal cells and the plant cells. And that was the botanist, and we cited as the botanist. And Swan was the geologist, and Meerut. Particularly this one, this is the phase we distinguish between the whole story concerning the plant cells and animal cells. And this one, if you talk about the whole genome and whole cellular level of the organization, which constitute the different level of the organization under the organ, tissue, and the cellular level. And this is the exceptions of the cellular level, because there are some of the microorganisms in the nature that was not made up of the cell. They are the acellular cells. They don't have any cells like the viruses and the other organisms. And this is the exception of the cell theory. What are the main postulates of the cell theory? If you talk about the cell theory, and the origin and the origin of the cells. This is the most important thing, that means cells exist for pre-existing cells. And the origin and evolution of cells. What are the origin, year of origin, where the cells originate from the different, and what are the evolution of cells? Definitely, this topic plays a very significant role. If you are talking about the fundamentals, basically the fundamentals things which is concerned with the cell and the cell life, origin and evolution of cell. And the first cell are all present-day cells. Prokaryotic and eukaryotic. This is the distinguished parameter in which, on the behalf of the assembly and the position of the cell orbitals, these cells are divided into two parts: prokaryotes and eukaryotes. Prokaryotes are the examples of the bacterial cells. Bacterial cells are the example for prokaryotes. And this is descended from the single ancestor. This is very important. And they descended from the single ancestors. And the first cell is thought to have arising at least 3.8 billion years. Again, I am saying this is just an average year. This is not a fixed measurement, fixed time. This is an average 3.8 billion years as a result of conversion of self-replicating RNA in phospholipid. And this is the present day prokaryote. What is the philosophy of the present day prokaryotes? This is divided into two groups, the archaeobacteria and the eubacteria, which diverge early in evolution. Because this is the extant form of the bacterial cells. Because if you talk about the bacteria, because it depends upon which the life of the bacteria is, because it depends on the existence of the years and what are the biological different factors which proves the age of the evolution. Eukaryotic cell, this is very complex and diverse form of the cells which exist in the nature differently on the intracellular level and the intercellular level. How could have evolved from symbiotic association prokaryotes, and this is also called the endosymbiotic theory. And this is the two main classes of the cells, prokaryotes and eukaryotic cells. This is the very fundamental thing about whenever we talk about the fundamentals in cell biology. This is the very keystone statement about the cell biology. That means cells are divided into two parts. One is the prokaryotic cells and the eukaryotic cells. And one more importantly, if you talk about the prokaryotic cells, this is a single statement, that is they have no nucleus. That means these cells have not two nucleus. These are simple structure. This is the bacteria, main instances of this group. And particularly when we talk about the eukaryotic cells, this is the major group of cells that exist in the nature diversity. They occur in the nature diversity in form of different forms, like the intercellular level and intercellular level. They contain nucleus, that means two nucleus is present in the eukaryotic cells. And particularly one important statement which this statement more strongly that they have more complex structures when we talk about proteins, fungi, plants and animals. And these are the proteins, these are simply the polymers of the polypeptides, and the polymers of nucleotides is called DNA. These are the main macromolecules. They constitute the whole genome of any cells, and any individual they represent differently. And this is again the group of organisms, fungi. This constitutes the different residue. They are also the part of the eukaryotic cells that exist in the nature. And of course, this is the plants and animals, again different parts of the nature that play a significant role for the maintaining the whole ecosystem. And this slide is also highlighting some important fundamental things, which is concerned about the sub-cellular structure. The sub-cellular structure that plays a very significant role for the organization of the different area of the organization, tissue levels, organ level, and cellular level. And particularly, this is the boundary of the cell. That means cell boundary is very important for the entry of desirable molecules and the undesirable molecules. Because cell proteins and all constituents play a very significant role in the screening of the molecules, either they have repel. And of course, cell membrane is the part of that assembly that is very significant role, and other is cell membrane, cytoplasm, and its other organs. Just like this is the master nucleus. This is the main organelle of the whole cell, and that's why this is the controlling agent of the cell. Nucleus is also called the controlling agent of the cell. And this is diagram is indicating the cell membrane, and they have a thin lateral elastic autonomous structure which envelops the cells, that means it covers all of the cell particles. And I already told you about the boundary, that is the protection of productive, and maybe permeability is very important. That means it depends upon the micromolecules and the signals they are producing from the different molecules. It may be any foreign antigens in form of antigen, in form of ion, droplet, hydrogen ion, any ion. It depends upon the signals that are produced from the inner core of the cells. And this diagram is indicating also the assembly in the variable part, the carbohydrate, food, glycoproteins, phosphatic biology, surface protein are present, indicating that it has a very complex symmetry that depends upon the functional role of the plasma membrane. Secondly, it consists of bilipid layer with embedded proteins that are in two forms, integral proteins and the peripheral proteins. In the peripheral area, the proteins are present in the peripheral area and the peripheral proteins, and those proteins which are present inside the cell, so integral proteins. Of course, this is cytoplasm and its organ, because cytoplasm plays a very significant role for the organization and the dimension of the different acids. Of course, in the protein synthesis, gene recognition, assembly of the DNA replication, multiplication, cells multiplication, and its organoids. This slide is representing in form of it have a very thick gel-like semi-transparent fluid that is found in the both plant and animal cells. As the diagram indicating, they have a different surface area, outer boundary, middle part in form of nucleus. As I told you, this is controlling agent, main controlling agent in form of nucleus. And the constituents part of the cytoplasm are cytosol, cell organelle, and cytoplasm. Otherwise, these are bounded by the plasma membrane and contains many organelles in a eukaryotic cell. And the cell containing membrane-bounded nucleus, as you not know that they have mitochondria, because this is the power of the cells. It plays a different role. Plasma membrane, I told you already, because it is a protective matter all in the nature, and the main permeability is very important factor which is concerned to the plasma membrane. Microtubules play a very significant role in the cell division. Centrosome, microfilament, lysosomes is very important part. This is also called the societal bags. It contains the hydrolytic enzymes. And there is the entry of any undesirable molecules that this is the synthesis, and their hydrolytic enzymes release, and the whole undesirable molecules from the cells. And likewise, the endoplasmic reticulum converts to two forms: a smooth endoplasmic reticulum and the rough endoplasmic reticulum. Secretory vesicles, vacuoles, cytoplasm, Golgi vesicles, Golgi apparatus, and it forms in the neural system. Golgi body, endoplasmic reticulum, and the lysosomes. Because arrangement is very important. Arrangement of microorganisms is very important, and the whole function depends upon the arrangement of the molecules and the function. And particularly, if you highlight the ultra structure of any organelles, then it indicates which types of the function, which type of function it performs, either the outside or outside from the cell, or inside the cells. So these again the chromatin network is again a very significant role, because chromatin network is the only the distinguishing factor that this is the main factor that distinguishes between the prokaryotic and the eukaryotic cells in form of the bacterial cell and the whole different cells. Because if you talk about the bacterial cells, prokaryotic cells, they don't have any chromatin network. They have the DNA and extra chromosomal molecule molecules are present in the prokaryotic cells, and like us, they have, because we are talking about the eukaryotic cells, they have the complete complex network that it performs for the identity and uniformity of the genome, because this is the main significant factor which is going to stay today, and whenever we are talking about the fundamental things of the cell biology. And of course, this slide is indicating the significance of cytosol. The cytosol is the aqueous part of the cytoplasm outside all of the organelles. It also contains its own distinctive proteins, and particularly it accounts for almost 70% of the cell volume, because volume is the significant role for the occurrence of the different volume. Genetic substance consisting mainly of cytosol atoms, organic molecules, salt, and water. Like us, chemically the cytoplasmic matrix is composed of many chemical elements in the form of atoms, ions, and molecules. And the rest, other than the nucleus, there are the different other organelles which is present in eukaryotic cells. It plays significant role individually. If you talk about the mitochondria, this is called the powerhouse of the cells. It has the whole quantity and the whole currency in forms of adenosine, triphosphate, diphosphate, and monophosphate. And time to time, when any cells require in the form of additive molecules, it provides to the whole part of the cell. And that's why this is also called the powerhouse of the cells. And if you talk about the ultra structure of mitochondria, they have the envelope system, envelope structure. That means intracellular matrix and the outer cellular space. There is a significant role. They have the different types of the proteins. It plays significant role in the different glycolysis, etc. and perhaps that maintenance of the whole power it controls. Like was endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, vacuoles. Each organelle performs its own different role for the unicellularity and particularly for the regulation and balancing the whole cell constituents. And each organelle is bounded by a lipid membrane and has specific function. So as I told you earlier also that the name of cytologist Patel firstly told us about the granular structure in the striated muscles, and mitochondria also called the powerhouse of the cells. This diagram is indicating, you can see here the inner membrane, outer membrane, and just like they have the different C phase and the M phase. If you highlight the C phase, it reflects the different structure. If you see on the M phase, it reflects the different structure. And in the fingers-like structure in reptiles, they have enzymes which perform the same role in the different systems like the glycolysis, and etc. electron transfer. When you talk about the length and the diameter, and this is the average length of each mitochondria, 5 to 12 micrometer. If you talk about the diameter, this is an average diameter of 0.5 to 1 micrometer, and this is the filamentous globular in shape. Likewise, if you talk about the endoplasmic reticulum, this is the network of tubular flat vascular structure in the cytoplasm, and this is an extensive network of the closed flattened membrane-bounded sacs systemically, like the coils. Whenever the stax coils are arranged in form of stax coils in form, and they have the nuclear core inside the nuclear reticulum, and they are attached to the smooth endoplasmic reticulum, and this is the space inside the tubules is filled with the endoplasmic matrix. And this is the difference between the smooth endoplasmic reticulum and the rough endoplasmic reticulum. In the smooth endoplasmic reticulum, ribosomes are absent. Meanwhile, in the rough endoplasmic reticulum, they have the ribosomes. And particularly the main difference that is distinguished between the smooth endoplasmic reticulum and the rough endoplasmic reticulum mainly present when we talk about the smooth endoplasmic reticulum, they mainly present in the lipid forming cells like adipocytes, interstitial cells of the testes, glycogen storing cells of the liver. These are the sites, these are the examples of when we talk about the function of cell organs. They have the adipocyte cells, they are present in the muscle cells, they are present in the leukocyte cells, etc. Likewise, left endoplasmic reticulum, they mainly present in the protein forming cells. This is the main important distinguishing factors we should not out. That is, they are mainly present in protein forming cells, just like the pancreatic cells, goblet cells, antibody producing plasma cells, and this is granules from the nerve cells, etc. And when we talk about the function is synthesis of proteins, particularly the main function is the synthesis of proteins aggregation, unsaturation of the fatty acids, muscles contraction. Here is commonly known as, this is also called the cytoplasmic reticulum in muscle fibers. Likewise, Golgi body perform different roles in the cell. And Golgi body is a collection of membrane enclosed cells composed of 4 or more stacks layer of thin, flat, and closed vessels lying near the site of the nucleus. Because this is the adjoining organelles concerned to the nucleus, and particularly most important significant point we should not inform if they consist of the multiple discrete compartments, and they consist of 4 functional distinct regions: cis-Golgi network, Golgi stack, and the trans-Golgi network. They have a main function that we do, and this diagram is indicating its position and the different performance according to cell to cell. And likewise, lysosomes. This is again the cell organelles, and this is the average diameter of 250 to 750 nanometer. And these are the irregular structures surrounded by the lipid membrane. And more acidic as rest of, as I already told about, they have hydrolytic enzymes. So that this is also called the suicidal glands of the cell. Because whenever the one position and the feedback condition comes in cells, and there is a subsaturation and some entry of the undesirable molecule either foreign antigen members inside the cell, then these hydrolytic enzymes release and then engulf forms immediately. And then one particularly most important factor is the indigenous step acidity. This is a real and where the pH is 5 is present by the action of proton on hydrochloric enzymes. Lysosomes are cell hydrolysis and they function best in the acidic pH. This is a very important point that they function best at the acidic pH. And these are the other cell organelles which is present in form of chromosomes. Their average diameter of chromosomes is 0.5 nanometer. And most importantly about the chromosomes is that the lipid bilayer membrane sounds which regulates what enters and exits. That is, enter or exist. Both are the both, entry and exist. And our chromosomes is responsible for that. And the most significant factor is associated with this organ, urate oxidase, specifically. And structure is similar to that of the lysosomes, but with a different composition. You can see they have a lipid bilayer, and they have the urate oxidase specific role in which growth enzymes secretes from the cells. And this is important points about the chromosomes. Chromosomes can be formed by the budding of by the division. At the time of the cell division, if by the budding process, we can be formed. This is, you can say, it contains the backbone of the whole cell division. Cytoskeleton. Cytoskeleton system. Can you know that?
नहीं है।